Welcome back, my dear students. Today we will learn about Phylum Porifera, the sponges. These are simple, multicellular, sessile aquatic animals with many pores for water flow that helps in feeding, respiration and excretion. They have a cellular level of organization, no true tissues and show asymmetry or radial symmetry. They reproduce asexually by means of budding or gamete formation, and they can reproduce also by means of sexual processes such as where larval stages are present. The classification of Porifera includes there are three classes: Calcarea, which made up of calcareous spicules like Sycon, Hexactinellida or Hyalospongy, which are also known as glass sponges such as Euplectella, and Demospongy, that is the largest group of poriferan or sponges. They have spongin fiber or silica. Their examples include Spongilla or Spongia. For memory purpose, the mnemonic, we can say that sponge are sessile. They have pores on the body. They have osculum, but there is no tissue grade organization. Glass or calcium made skeleton is there, and they have ecological roles. General characteristics. Poriferans are commonly called as sponges. The word porifera means pore bearers because their bodies are full of tiny holes. They are basically multicellular, but they are sessile. Once they settle, they never move again. Habitat. Most of the sponges are marine. They living attached to rocks and corals, but a few like Spongilla live in freshwater environment. Body symmetry. Poriferans usually show asymmetrical or radially symmetrical structures, and the body it can be cylindrical or vase-like. Their body wall is made up of two layers, so they can be treated as diploblastic, but the ectoderm and endoderm structure, which are found in true diploblastics like Nigeria and Ctenophora onwards, those are absent in poriferan or sponges. Among the two layers in their body wall, the outer layer is known as pinacoderm, which is made up of flat pinacocytes, and the inner wall is guanoderm, which is made up of flagellated guanocytes or collar cells. Between these outer and inner, that is in between pinacoderm and guanoderm, there is a jelly-like layer called mesenchyme, which contains amoebocytes, which are the wandering cells that do all the work in sponge body. Cell types. Poriferans or sponges are multicellular, we know, and that means they are made up of many cells working together. But unlike single-celled organisms, which consist of just one cell that performs all the necessary functions for their survival, like protozoa, but the sponges have a complex structure made up of many specialized cells like pinacocytes, choanocytes or collar cells, amoebocytes, archaeocytes, colencytes, etc. Now you should know the most important characteristic features of porifera, that is canal system. The water enters through tiny pores called ostia. It moves through incurrent and radial canal and finally leaves through a large opening called osculum. This constant water flow brings in food and oxygen and it removes waste. So it performs in three basic stages, that is for food collection, that is for oxygen collection or oxygen transportation, and the last one is the removal of waste. Three functions are performed by the canal system of sponges. Feeding. We know the choanocyte cells capture and engulf microscopic food from the water. So sponges are basically filter feeder because during feeding they filter the water. Filter feeders are feeding, their food is trapped as water passes through the ostia. The planktons can get trapped by the collar cells. Their archaeocytes basically ingest and digest food caught by the food substances, microscopic substances by the collar cell or choanocytes. Respiration and excretion both occur by simple diffusion, so no organs are needed. So in case of respiration, what happens? It happens by means of diffusion. It takes oxygen from water and oxygen diffuses into the cells, while waste products such as carbon dioxide leave the cells. This is the basic diagram showing the respiration in sponges. Skeleton. It is supported by either spongin fibers, which are soft, or spicules, which are hard structures made up of calcium carbonate or silica. These are the various kinds of spongin spicules found in the skeleton of endoskeleton of sponges. Excretion. The excreted product, mainly ammonia, which is released from the body through outgoing water current. It occurs like respiration. It occurs also by means of diffusion. The excreted products of the sponges, ammonia and other nitrogen containing substances. The excretion occurs through both the oscula and the surface of the sponge. Now, reproduction can be of two types in case of sponges: asexual and sexual. Asexual reproduction occurs by means of budding or gamule formation. Asexual reproduction occurs mainly by budding. This is the outgrowth arises at the base and bud grows in size, and but may retain attached to the present. After that, the bud may break out from the parent and get attached to the solid substratum. So this is the basic process of asexual reproduction of sponges. But the sexual reproduction occurs by means of internal fertilization. The choanocytes help transport of sperms to the egg. This is the sperm cell and egg shell. They will fertilize. They form the embryo. Then planktonic amphiblastula larva will form. Then the larva settles and attaches to bottom or other substances. This is the new sponge, and from the new sponge, it will budding by means of asexual reproduction also can form new sponge. So after the new sponge will go larger and mature, and it will be the adult sponge. Larval forms: amphiblastula, which is also known as calcareous sponges, and ragon larva, which is the larva of demospongy sponges are found. But except amphiblastula and ragon larva, there is another larva which is parenchyma. Amphiblastula is hollow oval larval stage. It is found in Scypha. This is the amphiblastula larva, and this is the parenchyma larva. It is solid oval or flattened larval stage. Now we discuss the classification scheme of poriferans, which was proposed by Rupert Barnes in 1994 in his book of 6th edition. Porifera can be classified into calcispongy, hylospongy, and demospongy. There are three classes: calcispongy, hylospongy, and demospongy. Calcispongy has two orders like Homocoela and Heterocoela. Hylospongy have again two orders, that is Hexasterophora and Amphidiscophora, and demospongy has three subclasses like Tetractylida, Monaxonida, and Keratosa. Tetractylida subclasses have three orders like Mixospongida, Carnosa, and Corystida, and Monaxonida has four orders like Hadromerina, Helicondrina, Poecilosclerina, and Haplosclerina. These are the tabulated form which will help you to remember to memorize the topic, that is the differences between calcispongy, hylospongy, and demospongy on the basis of spicules and canal system. This is also the differences which based on the habitat and shape, size, color of the sponges. This is the differences of calcispongy, hylospongy, and demospongy. Their different examples are here, and the diagram is also showing here. Classification: we know the class Calcarea or the Calcarea sponges. They are exclusively marine, prefer shallow coastal waters, less than 100-meter depth. They are small, around 10 centimeter, and cylindrical or vase-like structure. Their skeleton is made up of calcium carbonate, so these are called as Calcarea spicules. Their canal system can be asconoid, syconoid, or leuconoid. Only class showing all three types. Examples include Leucosolinia, Sycon, and Grantia. The orders include Homocoela and Heterocoela. Homocoela is the simple asconoid forms, example Leucosolinia, and Heterocoela is the syconoid or leuconoid forms, examples include Sycon, Grantia, etc. We'll further describe just after a few times the differences between order Homocoela and Heterocoela. This is the structure of Sycon, the example of Calcarea sponge. This is the table showing the differences between Homocoela and Heterocoela on the basis of canal system, body size, body wall, location of the choanocytes, and examples. So the hylospongy or Hexactinellida, which are also known as glass sponge, they are found in deep sea. That is about from 200 to 1000 meter. They are cup or vase shaped. Their skeleton is 6 red siliceous spicules forming a glass-like lattice. The conocytes lie in finger-like chambers. There is no true dermal epithelium. The examples include Euplectella, which is also known as venous flower basket. This is the images of Euplectella, and the second one is Hyalonema, which is also known as glass rope sponge. So the two orders of Hexactinellida are Hexasterophora, include spicules are hexasters such as Euplectella, and Amphidiscophora, where the spicules are amphidisc-like, example Hyalonema and Pheronema. The fun fact is the Japanese use Euplectella, that is, as a wedding gift, because inside this structure it leaves a pair of shrimps which symbolize the eternal love. This is the differences on the basis of skeleton in between Hexasterophora and Amphidiscophora on the basis of skeletal structure, body structure, habitat, and example. The third one is the demo spongy, which is a modern sponge, the largest sponge. 90% of all sponges, they are mostly marine, but they are fresh. A few are freshwater. They are brilliantly colored due to pigments in amoebocytes. Their skeleton is made up of siliceous spicules, not six-red, but spongin fibers are also present. In some sponges, both the spongin fibers and siliceous spicules are present. Their canal system is always leuconoid type. Their larva is parenchymal larva. Examples include Spongilla, Euspongia, Heliclona, Chalina, etc. Euspongia is the bath sponge. Subclasses and orders. The Demospongy class includes three subclasses like Tetracternellida, where tetraxon spicules are present but no spongin is there. Example Oscarella and Geodia. Subclass 2 is Monaxonida. Monaxon megascleres are there with their variable shape. Their examples include Tethia, Cleona, Helichondria, and Spongilla. The last subclass is the third one, which is the Keratosa. The skeleton only of spongin fibers, but no spicules are there. And examples include Euspongia or bath sponge and Hippospongia or horse sponge. This is that tabular form. They are showing the differences between Tetracternellida, not differences, there is a comparison between Tetracternellida, Monaxonida, and Keratosa on the basis of skeleton, habit, and examples. These are the another format which include the comparison between Myxospongida, Carnosa, and Coristida on the basis of the characters, skeleton, body structure, and examples. This is the further division. The comparison will help you to memorize the topic. The Hadromerina, Helichondrina, Haplosclerina, and Poecilosclerina can be categorized on the basis of their skeleton, body structure, and example. Lastly, Hartmann and Goryu in 1970 created a fourth class of sclerospongy for some coralline sponges collected from caves and the tunnels of the coral reefs in Jamaica. They discovered in the Jamaican coral caves by Hartmann and Goryu in 1970, but the leuconoid sponges with siliceous spicules, spongin fibers, and calcium carbonate skeleton. So they have similarities both with the calcispongy and hyalospongy. They are basically found in deep waters. Examples include Astroclera or Stromatospongia. Note can be modern studies merge them with Calcarea or Demospongy. So they can be included under Calcarea sponge or Demospongy group of sponge. So in today's lecture, we explored the phylum Porifera, the simplest multicellular animals, understood their general characteristics, and learned how they are classified based on the canal system, spicules, and body organization. We also looked at the example like Sycon, Spongilla, and Euplectella to understand their unique features and ecological roles. Remember, Porifera marked the beginning of multicellular life, forming the foundation for the evolution of higher animal groups. If this video helped you, don't forget to like, share, and subscribe for more simple and exam focused biology lessons. Thank you.